Stressed out? Yeah, I totally get it. Staring down a literal mountain of information, wondering how you're possibly going to memorize it all for Mr. Coy's final? Well, take a deep breath because you are in exactly the right place. Welcome to the Ultimate Study Explainer. We're going to take that massive pile of notes and transform it into a highly focused, high-yield bootcamp tailored specifically for this final exam. We're going to hit the exact structures, the specific pathogens, and, crucially, those classic trick questions that are practically guaranteed to show up and mess with your head. So grab your notebook, get focused, and let's conquer this material together. Okay, let's dive straight into our agenda. We'll be hitting the rules of the microworld, cell structures, the gram stain, high-yield pathogens, and finally, wrapping up with viruses and genetics. Starting with Section 1, Rules of the Microworld, Taxonomy and Nomenclature. Now, before we can treat an infection, we have to know exactly what we're dealing with, right? Understanding where organisms belong in the grand scheme of life determines exactly how they infect us, and most importantly, how we treat them. Let's contrast prokaryotes and eukaryotes. Eukaryotes are organisms with a true membrane-bound nucleus and organelles. This includes humans, plants, protozoa, algae, and, this is a big one for clinical treatment, fungi. Fungal and protozoan infections require completely different treatment approaches than bacterial ones. Why? Because their cellular structure is incredibly similar to ours. On the flip side, we have the prokaryotes, which lack a true nucleus. This domain includes bacteria, which reproduce by binary fission and cause most of our clinical infections, as well as archaea, which were discovered back in the 1970s and are thought to be the ancient ancestors of microbial life on Earth. Okay, you absolutely must know the strict formatting rules for binomial nomenclature, which were established by Linnaeus. Mark my words, this is a guaranteed test question. The genus is always capitalized, but the species epithet is never capitalized. Let me repeat that because it's that important. Do not capitalize the species. Also, both terms must be italicized or underlined. So when you write out Staphylococcus aureus, the S is capital, the A is lowercase, and the whole thing is in italics. If you see an option on the test like Streptococcus pyogenes with a capital P, that is a formatting error. Don't fall for the trap. Let's talk about microbial relationships for a second. The crucial point here is understanding parasitism. By definition, parasitism is a relationship where one organism, the invader, benefits while the host is actively harmed. And listen closely to this. All pathogenic infections involve parasitism. This is totally different from commensalism, where our resident skin flora benefits without doing us any harm, or mutualism, where both parties benefit. Think of mutualism like the algae and fungi that combine to form lichens. But if an organism is making a patient sick, it's operating as a parasite, period. Let's move to Section 2 and see how this builds. Cell structures and shapes, bacterial anatomy. We need to know what helps these microbes move, survive, and ultimately cause disease. First up, morphology. You have to be able to identify bacterial shapes under the microscope. Coccus means spherical or round. Bacillus means rod-shaped. Spirillum means spiral or corkscrew-shaped. But here's the classic exam trap you need to watch out for. Diplococcus. If a test asks you to identify organisms arranged as pairs of round cells, the answer is definitively Diplococcus. Don't let them trick you into guessing chains, which would be strep, or clusters, which would be staph. Diplococci are strictly paired round cells, a perfect example being Neisseria gonorrhea. Time for some rapid-fire structural facts. Think of the plasma membrane as the universal gateway. It's the main site for both active and passive transport into and out of the cell. For shear movement, bacteria rely on flagella or axial filaments. Then there's the pilus. Think of this little guy like a pipe. It's a hair-like projection used to transfer genetic material, like antibiotic resistance, straight over to a neighboring cell in a process called conjugation. But what about surviving extreme harsh environments? Well, organisms like Bacillus and Clostridium form highly resistant protective barriers called endospores. And finally, when our own immune system fights back, the final killing step, where digestive enzymes destroy an engulfed bacterium, is a process called degranulation. And that takes place strictly inside the phagolysosome. All right, let's do a quick call and response, just like your practice exam. We know certain bacteria have a protective outer layer called a capsule to help them evade phagocytosis. But here's the question. Which fungal yeast organism is definitively indicated by the presence of a capsule? Got your guess? The answer is Cryptococcus neoformans. It is the only fungal yeast that has this specific feature, making the capsule a definitive diagnostic indicator for this organism. Lock that name in your memory right now. Moving on to Section 3, Mastering the Gram Stain, Microscopy and Staining. I'm going to deliberately slow down our pacing right here, because this is absolutely crucial. The Gram stain is highly tested, and in the real clinical world, it's the critical first step for determining antibiotic sensitivity so a patient can get effective drug therapy before the full culture results even return. You have to know these steps in exact order. Step 1, crystal violet, that's your primary stain. Step 2, iodine. Step 3, alcohol or acetone, which acts as the decolorizer. Step 4, safranin, the counterstain. Gram-positive organisms have a thick wall that traps that crystal violet, so they appear purple or blue after the final staining. Gram-negative organisms, though, have a thin wall, so they lose the purple during the alcohol wash and pick up the red or pink color of the safranin counterstain. Now, what exactly is the role of iodine in that process? So, the crucial point is that iodine acts as a mordant. By definition, a mordant is a substance that increases the affinity of a dye to a cell structure. Basically, it helps bond the crystal violet directly to the cell wall. It does not rinse the stain away, and it certainly does not solidify the culture medium. It fixes the color. Aside from gram staining, let's run through a quick matrix of special stains. Malachite green is used primarily to identify endospores, which will, you guessed it, appear green. The acid-fast stain detects mycobacteria. India ink is a negative stain that makes capsules appear as this really distinct clear halo. But here is a key visual test to remember. Auramine O. This is a fluorescent dye used to visualize Mycobacterium tuberculosis. If you see a test question about a suspected TB specimen glowing bright yellow against a dark black background under a fluorescence microscope, your answer is unequivocally Auramine O. While we're talking about culturing and detecting bacteria, let's briefly touch on oxygen requirements. Obligate aerobes require oxygen levels of 20 to 21%. To put that into perspective, that is exactly equal to the ambient air we breathe every day. On the other extreme, obligate anaerobes are literally killed by oxygen. They require special reducing media to remove all the oxygen just so they can survive in a culture. Next up, Section 4, High-Yield Pathogens, the who's who of the exam. Okay, let's speed the pacing back up. We are going to rapid fire through the most frequently tested pathogen facts pulled straight from your practice materials. First up, a classic exam trap symptom question. A bull neck appearance is a characteristic sign of which type of respiratory infection? The answer is diphtheria. Anytime you see the words bull neck, immediately think diphtheria. Also, I'll give you a quick bonus memory hook for exotic organisms. If you see a question about a pathogen producing a distinctive violet-colored pigment, it is Chromobacterium violaceum. Violet pigment equals violaceum. Simple as that. All right, here are your guaranteed pathogen pairings for Mr. Coy's exam. Let's start with some highly unique culture requirements. Bacillus cereus stands out because it can only be cultured on media prepared with rice grains. Now moving to serious life-threatening conditions, Clostridium botulinum causes a really dangerous flaccid paralysis affecting the diaphragm. Watch out for any exam questions mentioning dented or bulging canned foods. Klebsiella pneumonia causes a horrific hemorrhagic necrotizing consolidation of the lungs, known as lobar pneumonia, which you'll typically see in older adults. Then there's Pseudomonas aeruginosa, which poses a uniquely fatal risk specifically for patients with cystic fibrosis. For pediatrics, just remember that Bordetella pertussis or whooping cough is incredibly dangerous for infants under 6 months of age. Finally, a very common fungal infection, Candida albicans. This is the organism responsible for thrush, which presents clinically in the anatomical area of the oropharynx. You absolutely need to memorize these associations. And finally, Section 5, Viruses, Prions, and Genetics, the non-living and mutagens. Let's focus on the non-living entities, genetic mutations, and wrap up with a little microbiology history. Now, what's really interesting about viruses is why they're classified as non-living. It's because they are obligate intracellular parasites that are entirely incapable of replicating on their own. They literally have to hijack a host cell to survive. When classifying them, the suffix virales, like caudovirales, designates the taxonomic group of order. The Baltimore classification system organizes viruses into 7 distinct groups. For instance, HIV is a group V RNA retrovirus. Also, don't let the word phage A confuse you. Phage A, including virulent phages that lyse infected cells during transduction, are simply viruses that specifically infect bacteria. Oh, and prions? Those are misfolded proteins that are actually the most closely related in concept to viruses, since both are non-living. Speaking of viruses, get this, striking statistic. Cytomegalovirus, or CMV, is an extremely common virus that is present in 60 to 70% of all humans by age 40. That is a massive chunk of the population just carrying this around.
Let's pivot slightly to genetics and mutagens. A mutagen alters DNA. We know ultraviolet light creates thymine dimers, which is a physical mutagen. But in a surgical setting, laser or electrosurgical plume, which is basically surgical smoke, is a major hazard. I really want to emphasize this. That plume is classified specifically as a chemical mutagen, which alters DNA-based structure and pairing. It is not biological, and it is absolutely not ionizing radiation. It is a chemical mutagen.
Wow, we have covered a massive amount of ground today, all the way from Linnaeus's naming rules to the depths of the gram stain and high-yield pathogens. Remember Joseph Lister, the father of aseptic technique who applied germ theory to surgery? He revolutionized his entire field with precision and careful practice. So my question to you is, are you ready to apply that same precision, trust your studying, and completely conquer this final exam? Review these concepts, trust the structure we built today, and you are going to absolutely ace it. Good luck out there.